Hello all, I welcome to the new video on our channel, that is Biology by Diya Chawla. I think you can see my achievements in this section. I have qualified a lot of national level entrance examinations like AIMS PG entrance for biochemistry. I was all India rank 3 in that. I have qualified CSIR NET, IIT JAM and GATE entrance examinations. So today we're going to discuss about a very important biomolecule, about the nucleotides that make our DNA. DNA is our genetic material. Okay, so today we're going to discuss about that genetic material, and we have to discuss that, who gave us the proof that DNA is the genetic material. First of all, we need to prove, right? In science, we need a proof. Ki bhai, what is it? How did you get to know that this is a genetic material? All right. So there were certain experiments that were performed by scientists to show that DNA was a genetic material and not the other biomolecules like proteins, etc., etc. So there was a lot of confusion amongst the scientists that, if protein is a genetic material or DNA is a genetic material. So there were they actually the genetic material had certain features, right? The genetic material should have certain features. So I have written certain features on the board that a genetic material should have, that, to make something a genetic material, you should have these many features. So it should be able to replicate so as कि उसके the progeny should get the DNA, right? When the cell cycle occurs, the division occurs, it has to go into the daughter cells also. So from one you make two daughter cells, so at least it has to replicate. All right. Then secondly, it should be chemically and structurally stable. Of course it has to be structurally stable. It should not be, very sensitive. It should not be prone to a lot of hydrolysis by alkalis or acids or whatever. It should be express itself in the form of Mendelian characters. Very important, as we know that Gregor Mendel had done certain experimentation and gave the gene as the term factors. Then there were certain scientists that coined the term gene, that, gene is something that is inherited from organism to organism, due to which there is a phenotypic expressions in the daughter progenies, right? Then it should be able to mutate, matlab it should be susceptible, it should actually accept the change, with the time it has to accept the change, and it should be able to mutate. All right. So these were certain features that the genetic material should have. So today I'm going to discuss the certain experiments that actually prove that DNA was a genetic material and not other biomolecules. As we know that proteins are extremely diverse, so scientists thought that why proteins can't be a genetic material. They're so diverse, they can perform so many functions, they can be inherited from progeny, from the from the mother to the progeny. Why is not that happening from the parent to the progeny? Why is protein not inherited? So there was a lot of conflict between the scientists. So there were certain experiments that were, performed by the scientists. So first was given by the Frederick Misher. It was not the proof, he just isolated, pus cells में से he isolated something called a substance called nuclein. Then it was further actually termed, there was another scientist, that was Altman, who named this nuclein as nucleic acids. Now there was this confusion that whether DNA is a genetic material or whether the protein is a genetic material. So first experiment was done by Griffith. So Frederick Griffith did the first experiment that coined the term transformation, and I will show you what was that experiment exactly and what was the conclusion of that experiment. So wait and watch for the first experiment. Now we are going to begin with the first experiment that was performed by Frederick Griffith, in which he coined the term transformation, and it was bacterial transformation experiment. So at that time he was actually a medical bacteriologist or microbiologist who was working on pneumococcus bacteria. So he found that there were two strains of pneumococcus bacteria, an R strain and an S strain. R strain was a smooth strain, R strain was a rough strain, sorry. S strain was a smooth strain. So smooth strain mein it had an additional outer layer which was absent in your R strain, which actually protected it from the immune system of the organisms. That actually was, that additional layer that actually gave the capability to the bacteria कि भाई immune system उसको recognize नहीं कर पाएगा. Now the immune system won't be able to recognize that bacteria. All right. And fight against that bacteria. So it gave it an advantage to that bacteria, the pneumococcus bacteria. So he actually did certain experiments. So first experiment that he did was bacterial transformation experiment. So in the first set of experiment, what he did was he took mouse and injected it with the S cells, matlab living S cells, living bacteria, that is that is pneumococcus bacteria, not streptococcus pneumoniae, pneumococcus bacteria, and he took the smooth cells. S cell means the smooth cells, the smooth bacteria, that means that had the additional S layer. So of course the immune system won't be able to recognize that bacteria, so it won't be able to fight against that bacteria. So what happened? The mouse died in that case. The mouse died in that case because immune system was not able to recognize that bacteria, and our immune system couldn't fight that bacteria. The mouse immune system couldn't fight that bacteria. So of course when living S cells were injected, the mouse died. When living R cells were injected, what happened to that bacteria then? So of course when living R cell was injected, the rough strain was injected. A rough strain was actually recognized by the immune system of the mouse, and it was killed, and as a result the mouse lived. Now there were other experiments that were done by Frederick Griffith. Now I'm going to draw them for you. So in the first set of experiments, he just injected the R cells and the S cells and saw whether the mice was living or dead. So in the first experiment, what we can conclude, that the smooth cell, the bacteria that had an outer layer, that additional outer layer, it was not able to survive. The mouse was not able to survive when it was injected with that. But when R cells were injected, the mouse was actually able to survive. The immune system was able to recognize it as foreign and was able to kill that bacteria, that R cell bacteria, and mice was able to survive. Now there was second set of experiment that was performed by Frederick Griffith that actually proved the transformation into the bacteria. So now I'm going to draw that second set of experiment for you. So in the second set of experiment, what he did was he took the first mouse strain, he put the heat killed S cells. Now heat killed S cells were killed. They were killed at 82 to 90 degrees Celsius, and they killed the S cells. The bacteria was killed. Then that killed bacteria was injected into the mouse. As a result, of course, the bacteria was killed. The bacteria couldn't do anything to the mouse, of course. So in this case, mouse actually lived. The mouse lived in that case. Now in the second experiment, very crucial experiment that actually gave the term transformation was this one. That heat killed S strain was actually mixed with the living R cells. So it was very surprising to the scientists that he has actually heat killed S cells, matlab S to killed hai, bacteria is killed, and living R cells are there. So expected, what was the expected? Ki, R cells are living, and that is actually injected into the mouse. So what was expected? That mouse should live, right? But in this case, the mice died. Why? So he concluded that there was something that moved from the S cells to the R cells and actually transformed the R cells to the S cells, and as a result, the mouse died. So what was that transforming substance? What was that transforming substance? At that time, they didn't know about that transforming substance. So he just gave the term called transformation. That is, there is a transfer of certain element from one cell to the another cell that was making the R cell actually infectious, and it could actually kill the mice. So what was that transforming element? So there was other scientists that actually performed certain other experiments like Avery, MacLeod, and McCarty, but they did it in vitro conditions. So they concluded with the Griffith's experiment. So they actually did another experiment which was performed by these three scientists. So I'll now show you what were these experiments, and they wanted to prove that what is this transforming material, what is actually inherited, what is actually going into the R cells from the S cells that is actually making it more infectious. All right. So now I'm going to show you, not perform, I'm going to show you the Avery, MacLeod, and McCarty's experiment. All right. So the next experiment was performed by Avery, MacLeod, and McCarty. So what they did was they created an in vitro condition in which they mixed the R-type bacteria, the R-type cells, with the heat-killed S-type cells, the DNA extracted from the heat-killed S-type cells. So what they saw was, when DNase was added, DNase is that enzyme that can actually cleave the DNA. Okay, so that can actually cleave the DNA. So in that case, when DNase was added, they saw that these R-type cells were not transforming into the S-type cells. That means DNA is that material that is actually transforming these R-type cells into the S-type cells. But when DNAs was not added, the actually transformation was occurring. So when DNAs was added, what was happening? The transformation was not occurring because it was hydrolyzing the DNA. That means DNA was something that was actually transforming the R type cells to the S type cells in the presence of the heat killed cells. That means DNA was moving from the R type cell, from the heat killed S type cells to the R type cells, and making it infectious and transforming it. That means the transformation element that was required in that case was actually the DNA. But now this experiment was in vitro. We actually need proofs in vivo, in the organism. So who performed that experiment? There was third experiment that was performed, and now I'm going to discuss about that third experiment that was performed in vivo. And it was the final experiment, final proof that concluded that DNA is the genetic material and not the proteins. So that conclusion was actually clear by that experiment. So let's show that experiment to you. So the third set of experiment was performed by Alfred Hershey and Martha Chase that actually gave the final conclusion that DNA and not the protein is the genetic material. So now what was that experiment? How did they perform that experiment? It is very interesting. Just keep your eyes open, ears open, and mind open and hear this experiment. Okay. So in this experiment, what they actually did was they took a bacteriophage. Now, what is a bacteriophage? Bacteriophage is actually a particle that can actually attack the bacteria. For example, we have viruses, right? So viruses are of different categories. So the viruses, the category of viruses that actually attack the bacteria, they are known as bacteriophages or the phages. There are viruses that attack us also, right? We are very much familiar with the coronavirus, right? So there are certain viruses that can actually attack the bacteria also, that are known as bacteriophages. Now, what is the structure of a bacteriophage? Let me draw it for you. So in the structure of the bacteriophage, it has this coat protein, this coat. There is a DNA. Right now I can't call it a genetic material. Why? Because till now it is not proved that DNA is a genetic material. All right. Then there is a neck. There are tail fibers which actually help in the attachment of this bacteriophage to the bacteria. Suppose this is a bacteria, this tail fibers are actually helping this bacteriophage to attach to this bacteria. All right. So this is the function of the tail fibers. So this is the coat. This is the DNA. This is the neck of the bacteriophage, and these are the tail fibers of the bacteriophage. So now I'm going to discuss the experiment, what they actually did. So what they did was they actually radio labeled the bacteriophage. They radio labeled the protein and the DNA component of the bacteriophage. So proteins are very much abundant in sulfur. So actually for the radio labeling of the protein, they used S35. And for the radio labeling of the DNA, as we had seen in the structure of nucleotides, in the structure of nucleic acids, that it is very much abundant in the phosphate group. So for radio labeling the DNA, they used P32 for the radio labeling of the DNA. For the radio labeling of protein, they used S35. For the radio labeling of the DNA, they used P32, phosphorus 32, phosphorus 32. Okay, so now we are going to discuss the Hershey-Chase experiment, what they actually did. Okay, so in the Hershey-Chase experiment, what they did was, first they did the radio labeling of the DNA. So these bacteriophages, what they do, they actually attack the bacteria. They inject their genetic material into the bacteria, and they actually divide inside the bacteria, and they make new phages like themselves. This is the mechanism by which the viruses actually replicate and form their kind of copies inside the body, right? They will enter their genetic material inside the cell. Then this genetic material makes its copies of itself, and they make copies of themselves, and then these copies actually burst the bacteria and then burst out, and then they come out of the bacteria. All right. Now what happened in the experiment, they actually they mixed the bacteria with the bacteriophages, and what they did was, when they mix both of them together, now bacteriophage will attach to the bacteria with its tail fibers, and what it will do, it will actually inject this genetic material inside the cells. It will inject the genetic material inside the cells. So it injected the genetic material inside the cells. So it was seen that S35 labeled was outside. That means the protein was outside, it didn't go inside the cell. The genetic material didn't go, the genetic material went, but the protein didn't go inside the cell. So what was the nature of that genetic material that went inside the cells? It was actually the P32 labeled molecule. And what was that P32 labeled molecule? That was actually the DNA. That was the P32 labeled molecule that went inside the cell. That was the nucleic acid that went inside the cell. So then they used the blender. By slightly blending the mixture, these bacteriophages were detached from the bacterial cells, and then they checked these bacterial cells, that what went inside these bacterial cells, what was that molecule that went inside the bacterial cell. And then they saw by this radio labeling experiment that not S35 but P32 entered inside the bacterial cells, the genetic material that is responsible for the inheritance and everything, for the formation of new progeny that was inherited into the new progeny, was actually the P32 ones. The new phages actually had the P32 labeled bacteria, right? The bacteria also had the P32 labeled thing, that biomolecule inside it. So this experiment finally proved that that proteins are not the genetic, they are not something that actually carries your genetic information, but DNA is the ones that carry your genetic information. All right. So in the conclusion, what they observed, that bacterial cells actually had the P32 labeled component inside them, and the new bacteriophages that were generated, they also carried the radio labeled P32 only. That means the ones that is actually inherited from the parent to the daughter, from the parent to the progeny, is actually the DNA and not the protein, because radio labeled phosphate was found in them and not the radio labeled sulfur. The radio labeled sulfur was found in the surrounding. What was found inside, what went inside, what was the genetic material that went inside, was P32 labeled molecule, that was your DNA. So it was the final proof that was given in vivo, and finally all the conflict was resolved, and everyone got to know that DNA and not the protein is your genetic material. So by at this note, ending, that DNA is the genetic material and it has all those features that we discussed in the beginning. Just go towards the beginning, see all the features that a genetic material should have, and you will see that yes, DNA is having all those features that actually prove that it is a genetic material. So thank you for watching this lecture, and I hope you actually enjoyed these all three experiments that were performed by Griffith. So in the conclusion, I want to conclude all these experiments. So first experiment was performed by Griffith. That was the bacterial transformation experiment. That actually Griffith saw that there was something that actually transforming the R strain into the S strain when they both were mixed. When the S strain killed S strain and the living R strain were mixed. So he saw there was something that was transforming the R strain to the S strain. But what was that something? What was that transforming element that was transforming the R to the S strain? Then there was experiment done by Avery, MacLeod and McCarty that showed in vitro, in the laboratory conditions, not in vivo, in the organisms. It showed that yes, DNA was that material that actually went inside, that transforming material that went inside the cells of the R-type cells, and they made it more infectious, and mice was killed as a result. Mice died as a result. Now scientists were not actually satisfied because it was in vivo, say in vitro, so they wanted something in vivo, something that was done in the body of the organism, like in bacteria. So it was the final concluding experiment that was performed by Hershey-Chase and Martha Chase and Alfred Hershey, that yes, DNA is the genetic material and not the protein, because the bacteriophages, the new bacteriophages that were formed as a result of the infection in the bacteria, they contained P32 labeled element and not S35 labeled, and the bacteria also had P32 labeled, and in the surrounding, what was remaining? The S35 labeled. That means that DNA and not the protein is your genetic material. And I hope you enjoyed all these three experiments, and I hope you continue with us and watch our lectures and enjoy them much more. So thank you. At this note, I end this video. Please do like, subscribe and share my channel. Thank you so much. Bye bye.